Welcome to today's lesson.
Let us have a question on flowering plants.
This specimen is a herbaceous plant. What does herbaceous mean? Answer. The plant is green and soft. Herbaceous plants are green and soft. Provide labels for III, axillary bud. Now, what are 4 functions of axillary bud? The answers are: axillary buds can develop into branches, flower, tendril, thorn. Fill in the blank spaces in the table below, making reference this specimen G. Good. So that is the table you have. You have the table and the answers. Leaf arrangement, stem form, leaf venation, type of leaf, etc. And those are the answers you have there. Now question, what is the importance of the study of the plant features listed? Answers.
For easy and uniform identification of the plant, the same identification by all scientists. Then 2, to know its characteristics so that it can be placed into the same group. It can be easy to be exploited. And 3, to protect it from extinction if the plant is endangered.
These are the reasons. Study
Specimen H
and answer the following questions. Provide labels for V, VI, VII, VIII, and IX. And the answers are: this is cuticle, this is palisade cell, spongy cell, lower epidermal cell, guard cell. Good.
Another question. Which part of specimen G is shown in specimen H?
Answer. Longitudinal section of the leaf is what is shown in
H. Another question. If there are hair-like structures in specimen G, where will you find them? The solution: hair-like structures can be found almost everywhere on the plant, herbaceous plant. So the terminal bud, the stem, the leaf, the axillary buds, and even the roots, the tips of the roots, you can have root hairs there, and they are hair-like structures.
If there are hair-like structures in
Specimen G,
what tool can you use to observe the hair-like structures? The answer: hand lens.
if there are hair-like structures in specimen H, that is the longitudinal section, where will you find them? Answer is, the hair-like structures, you will see that they develop from the epidermal cells, not stuck on top of the cuticle. All right? They are from the epidermal cells. And I've drawn them in already, as you can see. So that is it.
And if there are hair-like structures in
Specimen H,
what tool can you use to look at them? The optical microscope is the answer. Now, complete the following statements. That's the next question. When IX, right? When IX is turgid, dash. When IX is flaccid, dash. The answers are: when IX is turgid, this stoma opens. Here is a stoma, a hole here. Stoma. Many of them stomata. So when IX is turgid, the stomata opens. I have to explain that very quickly. This is a guard cell, this is a guard cell. In between the guard cell is a stoma. And this guard cell, this part of the guard cell is thick. The cell wall is thicker than the rest. So, and the guard cell is the only epidermal cell that has chloroplast. So, during the day, when there's light, photosynthesis takes place. There's sugar formed in here, so more water flows into the guard cells because sugar is in there and the concentration, osmotic concentration of the cell becomes high. So the guard cell, water flows in and the guard cells become turgid and the walls extend, and as they extend, these parts of the walls are thinner. So they extend outward like this and the stoma opens. When there is no light and no photosynthesis, the sugar there is converted into starch. So the osmotic concentration reduces, the solute reduces. So water flows out when there's no light. All right? When photosynthesis is not taking place, water flows out. And when water flows out, the cell becomes flaccid, soft. So it falls in and the stomach closes. So that is how the guard cell becomes turgid or flaccid. So we have another question. And the question is, if
Specimen G
is decapitated at point A, decapitation means the plant is cut off. It's decapitated at point A. What will be the effect on the plant? That's the question. The answer is dormant axillary buds near the tip will be activated to produce branches. So, remove this. These buds that do not produce branches at the tip, they will now be activated to produce branches. So once you cut the tips, more branches will be formed. Second question, what is the economic importance of decapitation of the shoot? The answer is, if we remove the tip and more branches are formed, it means more leaves will be formed. So economically, if the plant is used for the leaves are needed, there will be more greens. And then also, if fruits are needed, more fruits will be formed, increase yield. Good, so that's the importance. Now, another question is, briefly explain the biology behind the effects of the shoot decapitation. What happens? The biology behind it. What happens is that if this is the tip of the plant. This is the tip of the plant. This is the terminal bud here. This is the terminal bud here in detail, and there are leaves here. This is an axillary bud here. There's another leaf here. Now, at the tip here of the bud, there's what we call apical meristem. This meristem produces hormones, and the hormone it produces is called auxin. Produces auxin, indole acetic acid. The apical bud produces the hormones. The apical bud produces the hormones. The axillary bud also produces the hormones. The hormones flow down. The hormones flow down, and it causes the shoot to elongate. And a certain concentration of the hormones causes the shoot to elongate. Now, when the shoot is there, the axillary buds become dormant because the axillary buds also produce the hormones. So the hormones become too much. All right? So the axillary bud is dormant. But then when the terminal bud is removed, then the hormones produced in the axillary buds will be enough to cause the axillary buds to produce branches. So it will cause the axillary buds near the tip to produce branches. So they'll form branches. So that is what happens. Now I want to look at the last question. Here is the question. Looking at
specimen G and specimen H,
an experiment was done to test whether photosynthesis has occurred in the leaf in
specimen G.
Experiment was done to test whether photosynthesis has occurred in the leaf. Outline the steps to remove chlorophyll from the leaf during the experiment. Outline the steps to remove chlorophyll from the leaf. And the steps are: the leaf will be put in a test tube containing boiling water to kill the cells, to stop all chemical reactions in the leaf. Then the leaf will be removed and submerged in alcohol. The leaf will be submerged in alcohol, and this alcohol is put in a water bath. All right? This one is hot water. That's a water bath. All right? So the leaf will be lying in there. So that's the leaf. So you boil the leaf to stop all chemical reactions. You put the leaf in alcohol that is maintained in a hot water bath. The alcohol should not be put directly under fire. It will catch fire. And the leaf will be decolorized. Chlorophyll will be removed from the leaf, and the leaf will be discolorized.
Describe the state of the leaf after the chlorophyll is removed. Answer is: the leaf becomes white in color and stiff in texture or touch. Two things: white, stiff. Explain the state of the leaf after the chlorophyll has been removed. Solution: the leaf becomes white because the alcohol dissolves the green pigment chlorophyll and removes the chlorophyll from the leaf. And then 2, the leaf becomes stiff because the alcohol absorbs the water from the leaf. Good.
Now, last question. Indicate the parts of
In specimen H,
where chlorophyll will be. Indicate the parts of the leaf where chlorophyll will be removed. The answers are: chlorophyll removed from VI. There will be plenty of chlorophyll here. Palisade cells. There will be small amount of chlorophyll in the spongy cells. All right? And there will be chlorophyll in the guard cells. There will be no chlorophyll in the lower epidermis or upper epidermis, and no chlorophyll in the cuticle. All right? Why the upper epidermis and lower epidermis are transparent is to allow light to pass through. Neat. This is what we have for today. So we meet in our next lesson. Goodbye and thank you.